webinar series. My name is David Brown, the Sheep and Goat Specialist with University of Missouri Extension. And welcome once again to our MU Extension Sheep and Goat monthly webinar series. Before we introduce the guest speaker, please kindly note this following information. You may please type your question why our guest is presenting in the chat box. We're going to monitor that and make sure that those questions are addressed or answered once she's done with our presentation. In case you even have like the page number of the slide, you may want to include that so you can always go back and address those questions. And number two, please note that this webinar is recorded. And today's topic is abortion and respiratory disease in sheep and goats. And we have our guest speaker here today. She's very busy, so it's an honor to have her with us today, Dr. Maggie Ireland. She works with Wisconsin Veterinary Diagnostic Laboratory. She serves as a pathology and professional veterinary service aide. Dr. Ireland maintains a very strong interest in small ruminant health and disease, particularly infectious disease and immunology. Dr. Ireland, thank you once again for joining us today, and I'm going to turn everything over to you.
All right. Thank you for that introduction, Dr. Brown, and welcome everybody. I hope this is an informative session for all of you. And I did already notice a chat request for a PowerPoint presentation or a PDF version, and we will certainly provide that for you. And my contact information will be at the end of this presentation. I always welcome folks that have questions that would like to discuss. You are welcome to reach out via email to me at any time if you have follow-up questions that you think of after this seminar. So we'll get started. Let's start with the topic of pneumonia. So causes, we'll get right into it. Etiologic agents, so there are parasites or also referred to as lungworms, and there's 3 different species of those that can cause pneumonia in sheep and goats. And then bacteria that cause pneumonia include my favorite bacterium, Mycoplasma pneumoniae. I've done a lot of research and a lot of work on that particular mycoplasma. However, there are other mycoplasma species that can be associated with pneumonia and other diseases in sheep and goats, such as mastitis or poor joint health. Secondary, what I consider more, they can be primary, but often secondary bacterial agents are those Mannheimias. So that M stands for Mannheimia, and that's Mannheimia hemolytica, P, Pasteurella multocida, and the B is Biebersteinia traelosi. As I said, those are often secondary agents when an animal is compromised, when the lungs are compromised, either due to a primary mycoplasma infection or to a viral infection, which we'll cover next. So testing for parasites that can cause pneumonia, that's typically a Biermann fecal flotation technique. So these parasites get into the host via the intestinal tract, and they shed eggs that can be detected on the feces. So sampling for diagnostics, if you have an animal that passes away, that dies from pneumonia, we request that representative lung tissue, so that would be from the abnormal area of the lung, normal area of the lung, and then at the junction between those 2 areas of the lung, and I'll show you a gross image of an affected lung. We request that tissues be sent fixed and fresh. And fixation is usually in 10% neutral buffered formalin. That's something that your veterinarian, if you have one, should be able to provide. If you do not have the ability to fix tissue, fresh tissue being sent overnight delivery to your diagnostic laboratory of choice is preferred. So the quicker we get the tissue, the better shape it'll be in for us to make a diagnosis. So some of those viruses that I talked about that can impact often kids and lambs, but adults can also be impacted, include parainfluenza virus or PI3, respiratory syncytial virus, or referred to as RSV, and there's also an adenovirus that can cause or be associated with pneumonia in sheep and goats. Retroviruses are another type of virus that can be associated with pneumonia, at least in sheep. The most famous retrovirus is OPP virus, or ovine progressive pneumonia virus, also referred to as Medi-Visna virus, depending on what part of the world you're in. United States is probably the only place that uses this terminology as ovine progressive pneumonia virus. It's a very, very closely related virus. That's a retrovirus that will cause in goats arthritis and encephalitis, and thus it's called caprine arthritis and encephalitis virus, or CAEV. Pneumonia is not a typical presentation in goats that are affected by the small ruminant retroviruses. And I'll talk a bit more about ovine progressive pneumonia virus shortly. So here's an image that I promised to show. So this is lung tissue, and hopefully my my arrow is showing up, Dr. Brown. Can you see as I'm pointing things out? Does that show up on the screen okay?
Yes, we can see there. And Dr. Maggie, people say that your screen is split into two, like you.
It is split. Okay, let
Yes,
me try
thank
to
you.
share the other screen. I'm going to stop share just for a minute, and then I'll try to share. That's what I was, let's see. Is that better for folks?
Yes,
Is that the
yes.
single image? Okay, so it should be
Thank
bigger.
you.
All right, I shared the wrong screen. Um, so here is the gross picture that I promised of lungs. So the diaphragm would be back here. This is the front of the animal, so head would be up here. So this is a cranioventral distribution of pneumonia. So this darker red, not as aerated or not as soft, it's kind of firmer tissue. This is the common location for particularly Mycoplasma pneumoniae or bacterial pathogens that enter through the airway. So they enter through nasal or oral pharyngeal route, drop into the lungs, and this is typically the look that you will see. Sometimes you'll see white spots that are filled with purulent or pus material in the lungs also due to infections. But this really is a classic primary Mycoplasma pneumoniae infection. Symptoms that go along with this are typically maybe a snotty nose, a chronic cough. Some animals, you don't recognize anything is wrong at all. They're really good at hiding this. It's called a subclinical pneumonia then. But some animals that have Mycoplasma pneumoniae, their lungs are compromised. So if a more virulent form of bacteria, such as the Mannheimias or Pastorellas, drop into the lungs, it can be more highly associated than with death in an animal due to pneumonia. So a little bit about Mycoplasma pneumoniae. Again, it's considered a primary respiratory pathogen that can lead to those secondary pulmonary bacterial infections that can cause morbidity and mortality in your animals. This organism colonizes the respiratory epithelium, and when it does that, it impedes the little ciliary structures that typically clear out mucus and other bacteria that drop into the lungs. So on every breath in, bacteria drop into our lungs, my lungs, as I'm talking right now. But those little ciliary, typically when they're functioning properly, will take phlegm, and along with that mucus and phlegm, it'll remove bacteria that drop in as opportunistic pathogens. But when an animal is infected, and not all animals, it's not clearly understood why some animals handle Mycoplasma pneumoniae infections better than others. But with some animals that become compromised from this infection, they lose that ability. So then bacteria start to use the host lungs as a place to proliferate, causing a more severe pneumonia and more severe disease. This can lead to chronic pneumonia. Just Mycoplasma pneumoniae alone can cause a chronic pneumonia, but again, they're constantly being challenged by these other bacteria that are dropping in. And a common name for Mycoplasma pneumonia is, and it's typically in younger animals, is coughing syndrome or barn cough. It typically will happen, animals may be carriers of Mycoplasma pneumoniae with no signs. And then when we get to more humid months, or when animals are more crowded in a barn and air quality is poor, is at a lower quality, animals may start exhibiting signs of coughing, because those bacteria start to proliferate when the lung is further compromised by things like poor air quality, humidity, stressors. If you introduce new animals or due to overcrowding, you can also introduce different strains, if you will, of bacteria in that can cause sort of a coughing outbreak amongst your animals. You may have never had problems. I hear this from producers a lot. I've talked to several actually over the last year where they have brought in new animals. They had never had a problem, and they have seen this presentation that I give, and the signs and symptoms are pretty classic. Animals start developing a snotty nose, a cough when they're excited or trying to move around, or you'll hear just sporadic coughing amongst your herd or flock that you've never heard before. And then you become suspicious then that something's been introduced, such as Mycoplasma pneumoniae, with those new animals that were brought in. This organism was initially reported to be species specific, in that it would affect only sheep and goats, both wild and domestic sheep and goats. So bighorn sheep in the West, this is a big topic. If anybody follows wildlife issues in the West and domestic wildlife interface issues, this is a really big one that I had worked excuse me, that I had worked on for about 10 years before I made it back to the Midwest. Uh, so, but it's not just in sheep and goats as it was thought to be. I've identified it, and others have too, in deer species. I have, over the last few years, discovered that it's probably the primary cause of respiratory disease in captive white-tailed deer in particular. And there's a recent publication that describes it in wild white-tailed deer in the Midwest. There have been USDA studies. They're called the NAMS studies of different agricultural species. So the sheep study that was done back in 2011 identified that in large flocks, there's a pretty high prevalence of Mycoplasma pneumoniae presence. And then a 2019 NAMS goat study investigated the prevalence of this organism in goats. That data is still under analysis. When I was last at Kansas State Veterinary Diagnostic Laboratory and obtained a grant to test 8,700 nasal swabs collected from goats, and that's the NAMS goat study. It's currently sitting in APHIS's hands for all of the statistical analysis. So as you can imagine, it takes quite some time to test 8,700 nasal swabs in a research setting. And then the data analysis is quite extensive. So we're hoping that by this winter that will be completed. I had done a pack goat study looking at 587 domestic goats that were used for the purposes of packing, so hiking or for hunting purposes, companions in the woods. The tendency seems to be in goats that are, and it's true in sheep also, the smaller the size of the herd or flock, the slightly less likely that Mycoplasma pneumoniae is present. So it's larger flocks and larger herds that often have mycoplasma circulating. So it's easier to keep out, it seems, in smaller groups. And for goats, it seems that the kids and young under a year of age are the ones that shed at the highest level this organism and are the most impacted, where in domestic sheep, it seems like lambs often do not have it detected until they're over 8 weeks of age. So it's almost different. There's a difference there between those two species. And I will go to the next slide with that. So the challenges with this organism is it is exquisitely difficult to culture. And so it often goes underdiagnosed or undiagnosed as the underlying cause of pneumonia in species. And I think it wasn't until the early 1960s that this organism was even ever identified as the cause of atypical pneumonia or barn cough in New Zealand and Australia. And it wasn't until the 80s that it was ever first mentioned in bighorn sheep at all. So they had pneumonia problems for decades, and it was first identified on a culture plate in a bighorn sheep then. They went on for another 3.5 decades before it was identified to be the primary, one of the, if not the primary, one of the primary causes of pneumonia in wild sheep in the United States. So I would say the samples that I get, even using special culture techniques, 1 in 11 samples, I'll be able to actually get an isolate of this organism. So the best way for detection is PCR. So that's a molecular test looking for the bacteria's DNA and the presence of it. So Kansas State, while I was there, we developed with the help of their molecular team a very nice assay that will identify this organism specifically. So, and it's important when you select a diagnostic lab to submit to that they are specifically testing for Mycoplasma pneumoniae, as there are other mycoplasmas that are not considered pathogens, that if you use a general mycoplasma test, it'll show that it's positive. And those pathogens, those may not be pathogens that were detected. It could be the environmental mycoplasmas. So mycoplasma is a tricky one. And that was kind of a lot of information. So I encourage questions for further follow-up on that. The other thing about Mycoplasma pneumoniae, it doesn't have a cell wall. And it hides in the animal's mucus along their airways. So it's really difficult, if not impossible, to treat and clear the infection. So often the antibiotics, when they're given, people will report that the coughing has decreased, animals are feeling better, and then they will gradually start to develop that cough again and get sick again. And that's because the antibiotics often are treating those secondary bacteria that are falling into the lung. So it sort of holds Mycoplasma at bay when you treat with antibiotics, but to the best of my knowledge for the work that's been done, it is near impossible to clear Mycoplasma pneumoniae from an animal. The best option is you can try to keep it out of a flock. And as I already mentioned, this can be more difficult with large flocks and animals that go to fairs and shows. So there's a potential for exposure there also, in addition to bringing new stock in. So we'll move on to ovine progressive pneumonia. This was first described in Iceland, and where it was named Medi virus. Medi means dyspnea. So this virus causes difficulty in breathing. The animal, the lungs slowly become hardened, if you will, from this virus. And then Visna refers to the wasting syndrome that occurs in animals. And this is primarily due to, I call it the immune system being hijacked and the lungs being taken over by inflammatory cells. So when you have increased respiratory difficulty, that is a burden, metabolic burden on an animal. So they will lose weight. And again, this very closely is related to the caprine arthritis and encephalitis virus. And although reported, sheep or ovine progressive pneumonia virus infection in goats is documented, but it is extremely rare to occur. Once infected with OPP, it's a persistent lifelong infection. And sheep often remain subclinical silent carriers. They'll often live to be 5, 6, 7 years old before they ever, if they ever, show symptoms of having this virus. Pneumonia with OPP, again, that's the most, as the name would suggest, it's the most common form of disease. It is a prolonged insidious disease. Animals greater than 2, and as I just mentioned, most likely greater than 5 are affected clinically. So you may see an animal. My light just went out. Hang on, sorry. Must be sitting too still. Um,
Yeah.
um,
Thank you.
you'll see animals that will stretch their neck to try to get breaths in. The first animal that I experienced with this was a research animal while I was working with the USDA in Pullman, and I thought the animal was colicky. I thought there was some sort of GI problem because the animal was also kicking and stretching like an uncomfortable. I've noticed she was older, losing a little bit of weight, so not as in poor condition as the animal pictured here. And when we euthanized, the lungs were almost entirely very firm and taken over by the inflammatory infiltrates and scar tissue that occurs with this virus. So OPP can also be, I'm not going to talk about much more about mastitis other than to say it's also associated with a disease called hardbag in sheep. Transmission, virus is shed by those asymptomatic or subclinical infected sheep. Transmission is horizontal, so aerosolization or direct contact between animals. It can be shed in the respiratory tract secretions and in saliva, which they think may be the most important source of transmission of this virus. Urogenital, that's supposed to be tract, not trace. Tract, it can be shed in urine or vaginal secretions, also in semen, it's been identified. Close confinement obviously enhances transmission of the virus between animals. Vertical transmission, it can be transmitted through colostrum. It's thought to account for a lesser amount of the transmission within flocks, but it does occur. In utero transmission, so transmission from the dam to the fetus that they are carrying is considered to be quite low. I have a odd 12 there too. I will correct those two things on this before I send this out. So iatrogenic is also transmission, which means reuse of needles. So iatrogenic means that it's transmitted by human actions. So reusing needles is another common way of transmission for this virus. There's no treatment, there's no vaccine. Test and cull of infected animals is the best method to try to at least decrease the burden in a flock. Seroconversion can be detected by serology tests. The limitation to this is that an animal can be infected for upwards of 6 months prior to being able to detect it on serology tests. So serology tests are looking for antibodies that the sheep mounts against the virus. And this can take months because the virus very slowly replicates, and it takes a long time for the immune system of the host to recognize that there's a foreign invader there. There are commercially available tests to detect this virus are needed. I believe South Dakota has one of the only tests that I know of currently to detect, and I haven't worked with them much, so I do not know much about this test. Again, when I was at the USDA, our lab would run a PCR for this test, but we certainly need improved diagnostics to identify this virus earlier to help producers. It's advised to not use colostrum or milk from known seropositive animals. If you can segregate newborns from an infected flock, you can at least decrease the transmission that occurs due to contact. And focus should be on eradication, not control. So what that means is complete eradication from your flock if possible, rather than just trying to minimize. That's the feedback, if you want to investigate more, that the Ovine Progressive Pneumonia Society provides on their website, and their link is there. So small ruminant abortions. Hopefully these tables provide a good reference when I share the slides at the end for you. So there are a number of infectious agents that can cause abortion in sheep and goats. starting with Toxoplasma gondii, and then working through bacterial and the viral that you see listed here. Pretty ambiguous saying other bacteria, but there are other bacteria that may not cause outbreaks of abortions within your flock, but such bacteria could include Salmonella. Salmonella has been reported to cause abortion, or any bacteria that can move from the vaginal tract into the uterus during pregnancy, or in a bacteria that can be bloodborne and transfer. So it's not listed, but these are just the more common that we identify. Viruses include Cache Valley virus, border disease virus, blue tongue virus, caprine herpes virus in goats. So there are also non-infectious causes or toxin causes of abortion. Two plants to identify include locoweed and broomweed. I think these are more in the western states. You'd have to look at weeds specific to your regions, wherever you're signing into, to see if these are present. Nitrates, nitrate accumulating plants. This is not very common as a cause of abortion in sheep or goats. The rumen tends to break nitrates down, but it can occur. Levamisole is a dewormer, and it's been associated with abortion when given in late gestation. So we have to be careful when deworming during late gestation and what wormers we are choosing if it's needed while an animal's pregnant. So abortion, stillbirth, and weak kids can also be due to nutritional deficiencies, such as vitamin A, vitamin E, selenium, copper, and magnesium. And then iodine, excess or deficiency in iodine can cause goiter and can result in poor thriving kids. These kids are often born with a very large lump under their neck, and we often hear reported from producers that aren't familiar with goiter that there's a mass, and often folks will think that that's lymph node swelling in newborn kids, when in fact it's the thyroid gland lobules that become really enlarged in utero. So lambs will be born with very large swellings, if you will, or like a mass-like structure right at the junction of the jaw and the neck. So Toxoplasma gondii, we'll go into a little bit of detail, and I want to make sure that I don't go too long, so I save time for questions here. But I'll go into a little bit of detail on some of the common abortogenic agents, infectious agents. So Toxoplasma gondii, as many probably are familiar with, this comes from cats. They're the definitive host, but many mammalian species can be intermediate hosts, including humans. So it can be a cause of zoonotic infection. One of the places that Toxoplasma gondii is probably most mentioned is the reason that they recommend that pregnant women not clean litter boxes or take exquisite care if they do, because the infectious agent can be shed in the feces of a cat. There's indication and research that shows that cats typically only shed toxoplasma once in their lifetime, and then they often will not shed again unless they're somehow immunocompromised. But so during that shedding time, though, they can shed quite a few of the infectious forms of Toxoplasma gondii. The route of transmission is typically oral transmission route. So feline feces contain the infectious oocysts, they're called. So cats that defecate or in feed stuff and it's not recognized, so sheep or goats that can get oral route exposure that way. The infectious cysts live in tissue of immediate hosts, so animals such as squirrels or mice and rats that the cats are eating. That's how it transfers from cat to cat. It's been shown that once an animal is infected, a sheep or a goat is infected, or cows, that they can transmit the infectious form of the protozoa through milk So there are reports of infants, children becoming infected after consuming unpasteurized goat milk. There's a published study on that. Um, there, Toxoplasma gondii can transfer to the fetus through the transplacental transmission route. So a pregnant goat or sheep that ingests the infective stage of this organism, it will go into the bloodstream and it will transfer into the placenta and it will damage the placenta while there, and it will also enter the fetus. So it's typically most associated with abortions that occur in the third trimester or stillbirths of term fetuses. So this is one of the organisms that make it very important because sometimes the only lesions that we see and the only place that we see the organism is within the placenta. So it's really important when you're doing a diagnostic workup to submit placental tissue if at all possible. And I'll quickly go through at the end, sort of the tissue collection and what we like to receive to do a full abortion workup if we don't receive the whole animal or the whole fetus along with the placenta. So the next organism, Chlamydia abortus, it can be transmitted through semen of infected bucks or rams. It's one of the ways that it can be introduced into a flock or herd. Large numbers of this bacteria are shed in placenta, are found in the fetus and amniotic fluid and uterine secretions. So during lambing or kidding season, this can be transmitted to other animals via these tissue sources. Infection is by inhalation, so it can be aerosolized and breathed in or ingested, or again by venereal route from an infected buck, ram, or a ewe or a doe who carries it in the vaginal tract, in the reproductive tract. It does cross the placenta, and the abortions that are caused are typically during the last 2 to 3 weeks of gestation. So again, it's one of those late term gestation or can cause stillbirth. If animals are born alive, they're often born weak and have ill thrift. This is one of the causes of abortion storms that has to be ruled out. It can cause upwards of 60% of loss. It's commonly referred to as enzootic abortion of sheep and goats. And you may also see plus or minus concurrent pneumonia, arthritis, conjunctivitis, and retained placentas in the does and in the ewes. I have just does here, but does and ewes can also show these signs, and it may give you some indication that the abortion storm that's going on is due to this agent. This is also a zoonotic concern, meaning that it can infect and make people quite sick. And again, it's important to submit, I remind folks of this a lot because we often don't get placenta. It's important to submit the placenta for a diagnostic workup for diagnosing this agent also. Campylobacter, another cause of abortion storms. It's termed vibriosis for some reason. I think it was an old name for this organism. And it often can account for very high losses, upwards sometimes of 90% loss in herds or flocks that have this newly introduced. And it's often introduced when new animals that carry the bacterium in the intestine come into a herd or a flock that did not have this agent present before. We used to think of Campylobacter fetus as the primary agent of Campylobacter or vibriosis abortion storms, when in fact Campylobacter jejuni has by far overtaken Campylobacter fetus as the primary causative agent of Campylobacter associated abortions in the United States. Let me see if I have it. So this, I do have it at the bottom here. So again, Campylobacter jejuni is much more common than C. fetus in the United States. And this is really important for veterinarians, in particular, prescribing antibiotics to try to control abortion storms that are either thought to be or confirmed to be due to Campylobacter, is that by far and large, the vast majority of Campylobacter jejuni strains that are circulating in the United States are tetracycline resistant. So we have to pick a different antibiotic that is not a tetracycline family antibiotic. And I have a very close experience with this when I first realized this problem, even though it was first published in the United States in 2002 as being a problem with tetracycline resistance in Campylobacter jejuni. There was a flock out west while I was with the USDA in Pullman. They submitted fetuses. They thought that they were going to control it. They used a tetracycline as they usually had with abortion storms, and they were seeing no progress. And by the time we had diagnosed and had the genetic typing of the bacterium done to confirm that it was tetracycline resistant, they had lost 75% of their flock crop that year out of a flock of 3,000 animals. It was a devastating learning experience. So I talk about that as much as I can, the tetracycline resistance in C. jejuni. So Coxiella burnetii, more commonly referred to as Q fever, although Q fever is the disease that this agent can cause in humans because it too is zoonotic. Source infections, it can be transmitted in feces, urine, milk, vaginal secretions, semen, placenta, and amniotic fluid are big sources of this agent. So when the host, a pregnant dam, a goat or a sheep or cattle, dogs and cats are infected with this agent during pregnancy, it moves through the bloodstream, it gets into the placenta, it replicates like crazy. You'll get very high numbers. And another source of infection are ticks. It's just where it was actually first identified in the United States. I believe it was in Colorado when they were investigating a different infectious agent altogether. They identified this new bacteria, new to being identified in the United States, by looking at ticks and what bacteria they carry. So they think they play a minor role in transmission, but they are a source of infection for Coxiella burnetii. The scary part about this agent, I think what makes most people, and it's not something to be exquisitely fearful of, and a lot of people are, it is a reportable disease agent. So federally reported, and most states require that it is reported, but it has a very, very low infectious dose. So what that means is that this tiny bacterium, which can very easily be aerosolized when an animal it gives birth and the placenta comes out, aerosolization, it only takes one bacterium to infect another host, including humans. Typically in humans, you may have cold-like symptoms, aches and pains, waxing and waning fever. In the rare frequency, it can cause endocarditis in humans. The biggest fear in this being a zoonotic agent is that it can cause abortion or miscarriage in humans also. So caution with that. That's probably the biggest cautionary statement that I can give about this is for pregnant women that are working around ruminants that can carry this organism. In sheep and goats, it can cause late-term abortions, stillbirths, and weak neonates. So Listeria monocytogenes or listeriosis. This organism typically doesn't cause abortion storms. It can cause several or maybe just individual abortions. It is a common bacterium that is found in soils. So it's in the environment. It's considered ubiquitously in the environment, but it likes to proliferate best in spoiled hay or silage. It can also be shed in intestine, shed through feces in animals that carry it in the intestine. Infection with this organism is by the oral route, so ingestion. One of the sources, often in wet season, when you have feed out in a bunk that gets wet, it likes to propagate in that. And that's so animals eating maybe wet or spoiled hay or silage that has set in a trough, particularly in wet season times. Um, its transmission to cause abortion is it can enter the bloodstream of the dam, and it will move then to the placenta. And it, again, it can cause sporadic abortions in sheep and goats. And the fetuses, often the animal doesn't abort right away. The fetus will die in utero, and it will often be carried by the host for a while prior to being expelled. So you'll often see autolyzed fetuses being expelled. Or if if it does not kill the fetus, it can result in neonates being born very weak, or you may see them carry to term and be born, depending on when infection occurs. You can have stillbirths otherwise. So milk can be a source of infection. It can be shed by clinically normal carriers or clinically ill does and ewes. You can also see one of the other things, you may get retained placentas with listeria-associated abortions, or even if they expel the placenta, the organism can hang out in the uterus and it can cause a metritis, or secondary infections can also come in to that compromised uterus and cause metritis. And this is also commonly known in the human world for human illness due to it being a foodborne pathogen, so food poisoning. Brucellosis ovis, this organism primarily affects rams, and it will cause an epididymitis, so swelling and inflammation of the epididymis outside of the testes, and it can result in poor fertility then for that ram. It can cause abortion and perinatal mortality if it's transmitted to the young. You seem to be pretty resistant to becoming ill from this agent, and they often will clear infections if they do become infected, unlike rams who carry it and can develop more of a chronic epididymitis or be chronic carriers. And unlike most Brucella species, so when you hear brucellosis, like in bison or in white-tailed deer, this particular Brucella is not a zoonotic agent. So Brucella abortus, which is a zoonotic agent, it's been reported in sheep and goats, but it's really rare, and most states are completely free of this bacterium. And Missouri, I believe, is one of them. Michigan, Michigan and Wyoming are two of the states that probably have more of an endemic issue because it has entered into a wildlife species in those states. But this is a reportable disease agent, and depending on the state, Brucella ovis may also be reportable. Leptospira. So this is a bacterium with multiple serovars. One of the primary sources is urine from rodents or raccoon sources that contaminate water sources or feed sources for your sheep and goats. It can survive for months in contaminated feed, soil, and water. The maintenance source hosts vary between the different serovars. So Canicola is thought to be more of a dog, Icterohemorrhagia, more like raccoons, wildlife, Bratislava pigs. That's less important, I think, than just knowing that Leptospira species, any of these species can cause abortion. So the infection is by oral route, so contaminated feeds, water sources allow for the animal to take it into the mouth. It can then enter through broken mucous membranes or through mucosa of the eye. Bacteremia in the dam, so it goes bloodborne in the doe or the ewe, goes to the placenta, and it typically is associated with more sporadic type abortions or stillbirths. It also has been reported to be able to be shed in the milk, and it can also cause mastitis. I said I wasn't going to mention mastitis, and I fibbed. So hemorrhagic mastitis can also be an outcome of infection with leptospira. And this agent is also zoonotic and can make people quite sick. Mycoplasma species that can and have been associated with mastitis. These guys can also cause pneumonia potentially and can cause abortions. I just have them listed here. I've seen these much less likely and associated with abortion. So just I'll briefly touch on them and move on for sake of time. Cache Valley virus. This is an important one because this has spread across the United States. It's been found as far north here. I believe you have seen it in Missouri. This is a tricky virus. Source transmission is typically mosquitoes. A doe or a ewe is infected with little consequence. So they may be completely asymptomatic. You have no idea that they have been infected. However, this virus will go through the bloodstream into the placenta, and it can cause abortion, arthrogryposis, it can cause poor muscle development, and CNS lesions. So typically, if you have an animal that an aborted fetus that looks like this, twisted legs, open that brain and see if the brain is kind of a water-filled sac appearance. And you can be pretty certain in the United States that you're dealing with Cache Valley virus. Diagnostics can be tricky because the virus will come and go in 6 weeks even from the fetus, and the fetus may continue to live and then have these symptoms and be aborted late term, and you'll never detect the presence of the virus. So really the clinical picture of this virus is important and help giving a clue that you're dealing with Cache Valley virus. And this is what I was talking about. This is the brain. The skull has been removed and the cerebral hemispheres have collapsed because this animal has hydrocephalus. And I already covered that a little bit. That's a lot of detail that you can read through once I provide the PDF for you. But just this was from our UW research program manager. Thank you to Todd Taylor, who provided this image. A year ago, we had multiple reports of fetuses born. This one you can almost tell has more of a dome-shaped head and kind of the twisted legs or twisted back appearance. Not confirmed with PCR, but pretty certain this is a Cache Valley virus. Border disease virus, this is closely related to bovine viral diarrhea virus. It's a pestivirus. Carriers can shed virus in saliva, feces, urine, placenta or the placental fluids. Persistently infected sheep are a source of infection. So this is a virus that affects primarily sheep. But we can see persistent infected goats also, although it's very rare. You want to cull animals that are positive in your herd, that are PCR positive, and seronegative. So you all actually have to do a serology test and a PCR on blood to determine which animals to get rid of. So it's those persistent infected animals that are shedding this virus, but they think the virus is a normal part of their body, so they don't seroconvert, they don't develop antibodies to this virus. So you want to cull these animals. And then here's just a little bit of information about the abortions and what the dead fetuses or aborted fetuses may look like if they are infected before 60 days of age versus after 85 days of age. Sorry, I'm rushing through the end a little bit here, but I do want to give time for questions. And this is kind of a lot of information. So animals that are born weak, I will just cover this a bit, weak or otherwise normal, they may have a hair coat that looks like this, or they may be born weak and have a shaking syndrome because it affects their nervous system. So a few things to look for in lambs that are affected by this virus in utero. So BVD, there is reports of transmission between cattle and sheep and goats. You'll see pregnancy loss, abortions, stillbirths, fetal congenital malformations, again, in the brain. To differentiate this from that Cache Valley virus, you typically don't see the twisted limbs or the arthrogryposis. Neonatal morbidity or mortality, so animals may be born alive but born weak and die after. And again, PIs for this in goats in goats and sheep is extremely rare, as is just BVDV in general. BVDV and border disease are so closely related that the PCR that they use at Kansas State, I believe, is the same PCR to detect both viruses. So the molecular diagnostic test is the same test to detect both. So very closely related, those two are. Then we have blue tongue virus. Adult goats rarely show clinical disease. True sheep can show clinical disease. Both are relatively resistant though. Sources of transmission are biting midges, and live vaccines can cause iatrogenic infection and abortion. So for places that allow for vaccination, you have to be careful when you give this to the animal. Kids or the fetuses are often aborted. They'll be mummified. So the doe or ewe hangs onto those fetuses and they become sort of desiccated in utero. You'll also see congenital defects affecting the CNS. So again, this was where the brain should be, and it's really just two sacs of fluid that collapsed when the skull was removed from along the plane this way. So it's important to look at the brain. It can give some indication. But again, this virus doesn't typically cause that arthrogryposis or those twisted limbs. And we already covered most of this, so you can use this as a reference on how to differentiate with what you're seeing on an aborted fetus and what you should suspect for the cause of abortion. And I won't go into caprine herpes virus. I've not diagnosed it much. I don't know its prevalence. I always ask that when I talk about this virus. I just don't hear much about it in goats and have yet to diagnose this. I just know that it can cause, it can be transmitted by venereal route, and it can cause abortion really at any gestational age, but often it's late term. So there's my question about that prevalence in the US, and if anybody has ever really worked on that, because I have not found much, although I know it exists in this country. So here's some references that you can go back to also. This is what we do when we perform a necropsy on a lamb, and or a lamb or a kid, an aborted fetus, and it'll give you some ideas there. The best advice that I can give, because sheep and goat fetuses are small, just send the whole body to a diagnostic laboratory along with the placenta, if at all possible, so they can do a complete diagnostic workup. And then I have diagnostic plans the diagnostic plan that we go through when we receive an aborted fetus. And then just a plug for the Wisconsin Veterinary Diagnostic Laboratory. Out-of-state fees, we do charge out-of-state fees, but we provide all of the testing from the gross examination, the histopathology, and all of the ancillary testing for PCR and bacterial cultures for $160 under a cap fee. We do charge more for out-of-state because we're subsidized. We do require a veterinarian signature for submission. So this is where I'll put in a plug to try to find and work closely with your veterinarians on when you have disease issues such as abortion or pneumonia that needs to be worked up and treated. And with that, I'll stop because now I only have 7 minutes for questions instead of 15.
Oh, thank you, Dr. Maggie. Very informative. Wow. I'm trying to absorb everything you just said. Anyway, like we have quite some questions in the chat box. To
Great.
save time, Doctor, you want to please just go from the beginning and read those questions. Not everything are questions.
Let me there I can fill it up.
Yeah, Jasmine Das was asking about
Pull it up
period.
on the screen here. Pull it up.
Yeah.
You can fill the screen so I can actually read them. Um, you're welcome. I would like the PDF. Uh, Ruminant. They seem to have
Yes.
respiratory disease and passed quickly despite steroids and fluid. Yeah, I'm sorry to hear that. I've not diagnosed that very much, Jasmine. So thank you for that. I'm actually going to write that down and do a little more looking into mint toxicosis.
Yeah, from what I read, Dr. Maggie, I think it contains perilla ketones that can cause acute respiratory distress syndrome. you know
Interesting.
yes, so sometimes the young bucks are very predisposed to that. Sometimes, if the animal is more mature, to be eating that, just, it might not have any effect on that, or if there's a dilute effect, eating that with some other hay or grasses, but it's a common problem in goat production.
So pyrrolizidine, you said?
Perilla ketones.
Oh,
Yes.
yeah, I know very little about that, and I would bet we have that plant. I've not ever diagnosed that. So thank you for sharing that, Jasmine. Um, how infective are these diseases? There a reason to worry if animals are pastured versus barn kept? That's a great question. So, obviously, the closer an animal is to another animal, the more likely infectious agents can be transmitted. And it depends on which agents you're talking about. Respiratory agents. So not only are they more readily transmitted with close proximity, sometimes crowding in a barn, even if it would be sufficient area. It's often a time that air quality can go down too, depending on ventilation. So increasing susceptibility to transmission events and disease if animals are somehow compromised due to husbandry stressors, etc. But I would say pasture, so more spread out, less likely, but animals that are feeding in similar areas, if you have bunk feeder or salt feeder areas where they're congregating, certainly would increase transmission potentials. Um, uh, in Bovicera. So I'm not familiar with Bovicera. Um, Snotty noses can be caused. I can answer it this way, Jasmine, just to provide some feedback. Snotty noses, Bovicera maybe is like Bovishield. So vaccine, there are no vaccines for Mycoplasma pneumoniae. So if it's a primary upper respiratory infection with Mycoplasma, that will not help more than just keeping at bay the bacteria that it's used to protect from. And often animals will have only upper respiratory symptoms and no signs of true pneumonia. So no cough, just kind of that snotty noses. And the thought is those animals that have the snotty noses may very well be your shedders and source of infection. That's been investigated a little bit, particularly in bighorn sheep populations. Um, Split screen. Uh, What level of transmission is there from bodily fluids during the birth? So some of the agents that I covered are highly transmitted from birthing secretions in the placenta. So a few to really worry about. I'll put Coxiella at the top of the list, and then chlamydia is another one that is a big concern for transmission from placenta and bodily fluids or birthing fluids, so amniotic type fluids or uterine fluids. I do not know of mycoplasma, if you're referring to Mycoplasma pneumoniae being transmitted from external parasites like mites. Mycoplasmas like epithelial surfaces, which means they like the respiratory tract, and others like more so uterine tract infections. Once I don't know for those that can cause joint ill or arthritis, whether or not it's really been examined if mites and other biting insects can transmit mycoplasma, say putreficans, or agalactia? I don't know the answer to that, but that is a really good question on transmission of those organisms. Can we see toxoplasmosis? Will it show up? So it doesn't. You can see it in the oocysts, typically on fecal smears. Coccidia, they use floats, and I would have to ask our parasitology people if they can ever detect it on floats. Can we treat with antiprotozoals? Is it always result? It does not always. It's often sporadic, a sporadic cause of abortions. And I don't know if that has to do with the dose of the oocysts. They also say that animals that have been exposed prior develop an immune response, so they may not abort twice from this agent. That goes with humans also. In miscarriage, there is a recommendation that maybe humans that are fearful could get an antibody test to see if they have IgM, or IgG, so they know whether or not they've ever been historically exposed to it. They say you can do that for cats also, but not my expertise, so not giving clinical advice there whatsoever. But it can be treated with antiprotozoals. So I would get more information from your local veterinarian on that. Uh, does Campylobacter jejuni cause mastitis? Not that I know of. I have not heard of Campylobacter mastitis. Sharing. Oh, tetracycline resistance. You're welcome. Spread that tetracycline resistance word far and wide. So I can't give advice on antibiotics that can be used, but I can share a paper that looked at antibiotics with this group. I can attach it. When you have tetracycline resistance in the this is referring to Campylobacter jejuni tetracycline resistance. I can share a paper that I shared with a producer and veterinarians while I was in Kansas State that talks about other agents, other antimicrobials that have been shown to be effective when you have tetracycline resistance Campylobacter jejuni. I will definitely share that paper, Patricia. Is serum testing for Q fever reliable? I believe so. It was for me. I'm IgG positive and never knew I contracted it any time in my life, but I worked with sheep and goats since I was a little kid. So I don't know the accuracy. So false positive, false negative results for Q fever serology type testing in animals. I would have to look at the specific serology test to determine that. And I know there are PCR tests available for it, so vaginal swabs to see if you have carriers. And then we do the PCR on aborted tissues also. But I don't know for serology tests that are commercially available, what their sensitivity is. So I can't answer that specifically with all uncertainty. We had 3 goats lose their pregnancies. 2 were in the same pasture. Oh, yeah. If that happens again, it would seem like something was happening in that particular pasture, and whether it was toxins in plants or potentially a hotspot for listeria proliferation. It would depend on what the environment was like and what the pasture looked like and the feeding sources there. So hopefully this provides some information for you, Holly, for submitting whole fetus if possible for those losses. Curious about using ultrasound. Oh, Dana, for the ultrasound, out of my wheelhouse for sure. I would consult with, even if you can't find a small ruminant specific, I bet a bovine practitioner would be able to help with that. There's also a good source with American Association of Small Ruminant Practitioners. I know the listserv can typically only be veterinarians, but Perhaps look into that source. They may be able to provide some information on local producers for you. I know the topic of ultrasound has come up on that listserv quite a bit. Thanks. That most every goat in Texas has blue tongue because of midges. Yeah, blue tongue's pretty prevalent in the Southwest and West. Three were, yeah, that's a tricky virus. So one mummified. So the comment is we had a diagnosed BTV abortion, blue tongue virus abortion in Central Texas, and another goat bred at the same time that had one mummified and three live births that were perfectly healthy. That it's interesting why it would target one in the same pregnancy in the same dam. Yep, so the PDF will be provided through Dr. Brown for anybody attending.
I think the last one is from Mary, Dr. Maggie. Mary is also asking um, antibiotic therapies for mycoplasma.
Yeah, so resistance, so mycoplasmas are really tricky to treat with antibiotics in general. Mycoplasmas do not have a cell wall, and even antibiotics that target DNA replication or protein synthesis within are often hard to reach the mycoplasmas. So like Mycoplasma pneumoniae, we'll take that one for example. It hides out along the cilia and in the mucus. Some research indicates, I picture this little bacterium putting host proteins all over itself and masking itself. So if antibiotics that do reach the lungs at a high efficacy, that should be sufficient for clearing mycoplasma, often will not. So yeah, so it's not necessarily antibiotic resistance in the sense that the antibiotic can't kill mycoplasma. It's that it can't get to the site at a high enough concentration and reach all of the mycoplasmas to completely clear an animal of it.
Yeah, Dr. Maggie, we're going to take the last two from Jasmine and Tammy.
Uh,
Serum testing for K fever reliable.
I think I did is serum testing. Okay, so that was the one that we just answered above.
Yeah.
So the Q fever, I would have to look into the available serology tests to see what the sensitivity and specificity of those tests are. So I can't really speak to that. I mean, most diagnostic labs have to run testing to make sure that there is at least a reasonable accuracy with those tests. And I think I made that comment. They were accurate on me. I'm IgG positive. So for humans, I know humans have serology testing also. Was there another question?
Yes, the last one is from
There.
Tammy. Can you false positive on male with which be PCR possible? Do you see that, Tammy?
We used a more sensitive test though through TV, MDL, and got negative. So this is for Coxiella? Yeah, so it's part of the Coxiella.
Okay.
And false positives can occur. So I know of certain tests that um like for OPP, there were false positive problems with tests that were two different tests, one from Europe and one from the United States that they have really worked through to limit that false positive. You can get false positives depending on the serology test, depending on the inflammatory state of the animal. So if you have an animal that has high inflammatory cytokines, when you do that blood draw, for details that I don't want to go into, because they're very detailed as far as laboratory bench work. But it's suspected that some cytokines can cause increased binding of antibodies that maybe aren't specific for Coxiella, for example. I don't know that that's true for the Coxiella tests, but I have had that happen with OPP tests causing false positives. And I appreciate all of your time and listening. And I know that was a whirlwind. And I hope you walk away with at least one piece of information that is helpful. for your herds and flocks. And again, reach out if you have questions. Develop a good client-patient relationship with a veterinarian. I know it's difficult to find small ruminant veterinarians, and I hope that's something that this country really pushes forward to get more of. Just agricultural veterinarians in rural areas in general are really in need. So, and please reach out. I will share the PDF with Dr. Brown, and I will share the tetracycline resistance in Campylobacter paper with him also to share with everybody.
Oh, thank you, Dr. Maggie. We've been very, I think, say blessed to have you on today on this webinar session. Thank you for your time. Please, like Dr. Maggie said, we're going to send the recording and PDF version to you with the last paper on tetracycline. So we want to thank you once again for joining, and we'll see you next month. All right, bye.
Thank you all.